Again, I'm Rachel Plunkett, and I am a graduate student at Oregon State University, and I'm also an employee at the Columbia Basin Agricultural Research Center. And so today I'm going to be talking to you about nematode interactions in wheat cropping systems of the semi-arid Pacific Northwest. And I'm just going to attempt to advance here. There we go. Okay, I don't know if that was me or if that was you, Carol. That was me. Okay, looks like it's working now. Today I'm going to be talking to you about two separate experiments that are looking at nematodes in the wheat-growing region of Eastern Oregon. So the first is the Resilient Dryland Farming Alliance, or RDFA, and then the second is a long-term experiment of nematode research that we're doing at the Columbia Basin Agricultural Research Center. So to start out with the RDFA, it is a grower-led initiative in the wheat-growing region of Eastern Oregon. It is data-driven, and it looks at replacing traditional fallow with cover crops. So traditionally, when you have the winter wheat, summer fallow system, you have one wheat crop that is yielded per like 14 months. And fallow is a water storage solution in that it keeps that soil retained water available for the cash crop. However, it does increase soil erodibility and it can reduce soil carbon. On the other hand, cover cropping has been shown to improve soil health, soil organic matter, pH, and plant available nutrients. For the RDFA, we deployed experiments at two separate study sites. We have an intermediate rainfall site in Umatilla County, Oregon, and a low rainfall site in Morrow County, Oregon. We have 10 treatments total, and those include both fall and spring season crops of winter pea, winter lentil, fall mix, purple top turnip, spring barley, phacelia, yellow mustard, common vetch, spring mix, and a fallow control. These treatments are all replicated 4 times, and they are arranged in a grid-like pattern, as you can see in the photo there on the right. Within the RDFA, we wanted to fill the following knowledge gap of what are cover crops' effects on nematodes. And we asked the research question of how do non-irrigated cover cropping treatments in the semi-arid Pacific Northwest interact with the root lesion nematode Pridelanchus neglectus populations. We hypothesized that differences in nematode abundance will vary between location and between treatments, considering a cover crop's effect on soil health. In order to answer our research question, we deployed our fall and our spring seeded cover crops. They were planted in fall and terminated in spring, respectively, for the fall cover crops. And the spring seeded cover crops were planted in spring, earlier in spring, and then terminated later in spring. Our soil samples were collected from each study site at a 30.5 centimeter depth, which is about 1 foot. And then from these soil samples, our nematodes were extracted and then counted per 100 grams of soil, but they were reported per 1 gram of soil in all of our figures and our results. And then in the photos on the right, you can see our team checking the seed depth in the lower photo and then taking soil samples in the upper photo. Moving on into our results for the RDFA, this figure shows the nematode count for both Morrow and Umatilla County, with Morrow on the left and Umatilla on the right. The y-axis is going to be P. neglectus per grams of soil, and our x-axis is the cover crop. So we did see that there is a difference between locations, with our Morrow site having more nematodes for all treatments than our Umatilla site. Also in Morrow specifically, there were more nematodes in the purple top turnip treatment than any others. For Umatilla, there were no differences between the treatments. Now, I'm going to move on into our second experiment, which is at the Columbia Basin Agricultural Research Center, which is located in Umatilla County near Pendleton, Oregon. We focused on a long-term experiment at Seabark, which is the crop residue experiment. And this is a 2-year rotation of winter wheat and fallow with a moldboard plow tillage throughout. There are 9 treatments total, and these treatments include different nitrogen rates, burning or not burning, and then additions of natural fertilizers, and then no additions that are acting as a control. So the biggest difference between this experiment and the RDFA is that we are looking at root lesion nematodes, which are probably the most commonly found nematode in our region that are causing the most economic losses to growers. However, we also considered the entire nematode community. And in our soil samples, we found stunt, spiral, bacteria feeding, and fungal feeding nematodes in addition to the root lesion. For this experiment, we really wanted to look at how crop management can affect nematodes at seabark long term, because this is an experiment that has had consistent management for many years. So we asked the research question of how do cropping treatments in the semi-arid Pacific Northwest affect the soil nematode community? And we hypothesized that differences in nematode abundance will vary based on how cropping treatments affect soil health. We considered all of the treatments within the crop residue long-term experiment, and that includes a control, which is going to be no burning and no additions, a pea vine, manure, spring burn, fall burn, no burn, and then a spring burn with a nitrogen addition. In order to discount any edge bias, we did not consider the two controls that were on the very edge of the experiment. We took soil samples from these treatments, and nematodes were extracted by liquid suspension from 250 grams of soil, and they were counted by individual trophic level. Our samples were also processed for total carbon, total nitrogen, and pH. Looking at the results for this experiment, this figure shows the treatments on the x-axis, and then you have nematode count on the left y-axis, and then the percent soil organic matter, total carbon, and total nitrogen on the right y-axis. And the biggest thing that I want to point out here is where you have the peak in soil organic matter, total carbon, and total nitrogen. You also have fewer root lesion nematodes, and there's greater nematode diversity overall for these pea vine and manure treatments. So here's kind of the big question that I want to talk about today is, what do these results tell us? Well, starting with the RDFA, results indicate that cover crops do not exert short-term impacts on nematodes. We do have that location difference between the two study sites, but we think that these differences could be related to climate because, again, we do have a low rainfall and an intermediate rainfall site, so there are climatic differences between the two. And because of that, we can't really make the connection specifically between cover crops and nematodes because we're not really seeing any differences within treatments at either site. And then we can also conclude that multiple years are going to be necessary to observe trends within this experiment. And we do have another year's worth of data that we're going to be working on to see if the trends that we already see continue. For the long-term experiments, our results indicate that nematode abundance is directly related to soil health. We see that higher total carbon, total nitrogen, and soil organic matter supports greater nematode diversity. And within that greater diversity, that means that there's more competition between resources between our plant parasitic nematodes. And we also see that treatments with less nematode diversity are favoring those harmful root lesion nematodes, which again are the most common for our region and are causing the most economic loss. So overall, there are two big takeaways here. The first is that cropping treatment and a crop's type effect on soil health may be the greatest influence to regional nematodes, rather than just crop type alone. And growers can use this knowledge to better understand how crop management is affecting nematodes. Moving on into my acknowledgements, I'd like to thank my co-authors on this project and our research assistants and technicians who have aided in fieldwork, lab work, and analyses. I'd also like to thank the Columbia Basin Agricultural Research Center, the USDA Columbia Plateau Conservation Research Center, the Oregon Wheat Commission, our RDFA NACA funding, and Starvation Farms. And of course, I'd like to thank all of you for coming here today. When I was out at our Morrow study site, I did find a soil crust that looked kind of like a heart. And so I thought it was a great picture to say thank you for coming. And I'm looking forward to chatting and answering any questions you may have.